All right, hopefully you can all see my screen, and I'll go ahead and start with my presentation. So as Alejandra mentioned, my background is in material science engineering, and most of the work that I'll be presenting today is actually from my work at Harvard University's Wyss Institute, which focuses on biologically inspired engineering. And currently I'm at Counterculture Labs, kind of as a mentor and as a knowledge resource while I'm working on my startup, which is making tropical wines with Ikenga wines. So hopefully, as we go through this presentation, you'll start to see kind of how all these worlds start to intersect. So something that you've heard about probably for the majority of this Congress, if you've been attending the other sessions, is this notion of biotechnology, and many people's approach to it being trying to use living systems to produce products that are desirable for our needs. And I kind of, through this presentation, would like to shift this discussion towards actually learning from living systems of how to better design and better approach problem solving. So I'm coming from the material science world, and in the material science world, we're looking at how does structure kind of result in these interesting properties that have really unique performance, and how can we start to shape the processing that we do for these materials to get these new properties. Something that I personally look at and think about a good amount is thinking about human history and human through materials advancements. So kind of starting off with the Stone Age, we're working with stone tools just to kind of like these naturally found objects and just trying to shape them into useful shapes. Then we kind of start moving more into natural metals, not doing refining. Then we start to learn how to refine, and moving forward, we get into more chemistry. How do we manipulate small molecules? How do we start isolating them? So this is kind of where my foundation is originally in chemistry, chemical engineering. And then starting to control these small molecules and react them to form larger molecules such as polymers. More recently, we're kind of entering this age of advanced materials. So how we put the materials together starts to matter. So you're not just dealing with a uniform material like a polymer that has kind of a uniform property of elasticity, but now maybe you're putting metal inside of a polymer, and that's going to behave a lot differently. And something that has been touched on very thoroughly in this Congress is the biological revolution. So, each of these stages, we're learning how to work with different length scales and learning how to do more and more complex design tools. So when it comes to biology and this biorevolution, we're making these chemical changes to DNA per se, and or maybe the environment that some living system is existing in, to then have this cascade of responses. So what I would say right now is we have a lot of different tools at our disposal. We have good ways of seeing at the molecular length scales, at the atomic length scales. We're starting to have better control over our fabrication techniques of being able to precisely deposit materials down to the atomic length scale. So this is where you get into computer chips. And I would argue that now we need to learn how to use these tools more efficiently. And I'm hoping that this presentation today will give a good outline of some new design strategies that we can learn from nature. So today our teachers per se will be, we have three different modules that I'll try and rush through. The first being learning from this carnivorous plant and learning a little bit about how do we design wedding interfaces, just looking at this plant and how it behaves and how it survives. Then we'll move on to stripes. So stripes and patterns are found throughout the animal world, frequently through the hide or the skin. So I'll be kind of talking about that and what we can learn from that and how we can start applying it to make our own patterns. And then going into color if we have enough time. So it'll kind of be a teaser, little rush talk on the color side, but hopefully you can stick with me. So diving into the wedding interfaces, we'll start with this carnivorous plant. The plant typically grows in nutrient-poor soils and is found in high humidity environments. typically in Southeast Asia, but there are a bunch of different varieties throughout the world that often are in similar environments to this. And the reason that they're carnivorous is that they grow in these nutrient-poor soils. So it needs to get some of these nutrients from animal sources or from some other source. If we pay attention to the peristome, which is just this lip at the edge, we could see that when it's dry, you don't get, the ants can just kind of walk all over the lip. But when it's wet, they end up falling into the digestive cavity and then getting trapped and being consumed. So we see that the high humidity is pretty important. And throughout this, the next few slides, we'll be diving into exactly how this works and then how can we make a synthetic analog to solve our own slippery problems. So if we zoom in on the peristome of the pitcher plant, we start to see that there are these different length scales of roughness that we find. And when dry, you can imagine that an ant per se would just encounter a solid surface and would be able to walk on it. The material that this lip is made of is very hydrophilic, meaning that it likes water and it attracts water and can trap it. So when it's wet, it's able to almost be like a sponge and absorb water and allow a thin layer of water to be presented at the interface. And this may sound somewhat familiar to some problems that you may come across in your everyday life, which is maybe you're driving and it's raining outside and your tires start to slip and you hydroplane. And this is a really similar thing to what the ants are experiencing. So when it's dry, they can walk on the solid surface, no problem. But when it's wet, they're hydroplaning or aquaplaning directly into the gut of the pitcher plant. So this is something really interesting. And if we can learn from this of how can we trap a liquid layer that will cause something to not stick, we may be able to solve a lot of sticky problems that we have. Some of those, there's so many, but just to name a few, some of them may be your glasses getting dirty, or maybe you have, there's heat exchangers or pipes that end up getting blocked with life, just biological growth. Or could be corrosion. Maybe you drop something on your clothes while you're eating and you get a stain. So that would be a great example of how we would like to prevent the food stuff from sticking onto your clothing. Even in your refrigerators, if you look in your freezer, the freezer goes through these defrost cycles. So it will be staying cool, ice will accumulate on the heat exchanger, and it needs to melt it. So what if we can prevent or at least reduce the ice from sticking? And something that I'll really focus on this presentation is how can we prevent living organisms from sticking to surfaces, which is much more difficult than these kind of other cases. So in one example, there's a ship holes, and then another may be in a biomedical implant. So if you're putting a catheter into your body, you don't want it to get infected. That means that you don't want bacteria to be able to migrate along that catheter and into your body. So if we start just by thinking of the physics of how does the pitcher plant trap water on the surface, we can just start by trying to understand how does water or any other fluid, how can we start to characterize that interacting with the surface? The Young's equation is able to essentially give us a description of this balance of forces when we're at the interface. If we were looking at the interface, let's say this contact point right here, where you have three phases that meet. You have the solid, you have the liquid, and then you have the gas. And there is this balance of forces where you have the solid gas interface kind of exerting one force in one direction, the solid liquid kind of exerting a force in another direction, and then this balancing force, which is the liquid and the gas going in the perpendicular direction. And the balance of these forces ends up giving you a contact angle, an apparent contact angle, which we're pretty familiar with when it comes to just our day-to-day lives. We often see things that may wet, and if you maybe apply soap when you're washing your dishes, after you apply soap, if you put a little drop of water on some glass, you'll notice that it wets a lot differently. So that may be a metric of how clean it is. And getting into the cleanliness, getting into the surface, we start to think about the surface chemistry. So in some instances, you may have a hydrophobic surface, which will present as something that may be maybe greasy or something that doesn't like to mix with water. But if you were to, let's say, clean that surface and then maybe functionalize it by putting something that's like OH group, so maybe it's metal hydroxyl kind of bonds that are on the surface. You could do this through plasma cleaning or various other ways, silanization. Then you can change a surface that was hydrophobic into a surface that's hydrophilic. And this again is sort of described and captured by this Young's equation. Uh, these are for ideal surfaces, and surfaces are frequently not ideal. So if you start adding some roughness to it, if we just kind of conceptually think about it, if you have a surface that is hydrophobic, and then you start to increase the surface area of that, just roughen it, start to increase the surface area that the liquid's interacting with, you'll go from a hydrophobic to a even more hydrophobic system. So if you start to design your surfaces in really kind of precise ways or really unique ways, you could start to now increase how much water doesn't like the surface just by changing the roughness with a given chemistry. You can also do the converse. So if you have a surface that likes water and you start to roughen it, now it's going to really start to attract water a lot more. And this is kind of what we start to see in in nature. So two examples of kind of opposite extremes, where we have a lotus leaf, or maybe a different kind of leaf that you may find in Guatemala, but a leaf that will repel water. Maybe you just see raindrops kind of shedding off of it very easily. And that is done through this really small roughness that the leaf is able to produce. And oftentimes this roughness is on multiple length scales. So you kind of have this micro length scale and then a nano length scale. And if you do this really, really well and really precisely, and you don't have any defects, then you're going to get to a contact angle that's close to 180 degrees. So essentially the surface is completely repelling the water. If you have any defects on there, then it's going to be reduced. So in nature, in this leaf per se, if it gets scratched or if it gets bent, or just doesn't produce, uniformly, you're going to get some amount of pinning and sticking. And again, the opposite is true if you make a super hydrophilic surface, so a surface that really, really likes water, like in the pitcher plant case. So how can we, I guess if you have this nanostructure and this microstructure, you're able to, that really likes water, you're able to get almost perfect wetting, and you get something that's just a conformal coating of this liquid onto the surface. So this sort of explains some of these natural phenomenon that we may experience. And in practice, we could see that, on the left, we have biological systems, and on the right, we have an inorganic system we made out of titanium. So just talking about super hydrophobic and being able to prevent water from sticking, we're able to do that just through the nano and microstructuring with the proper surface chemistry. So we could see that in the biological case, there's a little bit more sticking. It's not as perfect as with our inorganic case. But with the inorganic case, we have nearly complete repulsion, and we're working with the material that's more robust than what you find in natural systems. We're working with titanium. So if we go back to the ant case, in the ant, on a super hydrophobic surface, would not be repelled. It wouldn't be anything special for the ant. It would just keep walking because it's in contact with a solid surface. If we have a trapped liquid layer, like we do in the pitcher plant case, then it hydroplanes. And now we could start thinking about, okay, we understand these very simplistic systems that can repel water, but how about more complex fluids? How about blood, let's say, or bacteria? Bacteria are so small, they're often smaller than the features that we're going to be designing. So they'll be able to just infiltrate into these cavities and grow at will. And then maybe, marine life, that may be a really, really complex system where you have bacteria, you have mussels, you have all these things from different length scales and different attachment strategies vying for position on your surface. So in the case of, let's say, blood, if you have a water film that's trapped on your surface, water and blood can mix. So that's, just trapping a water surface isn't going to be able to prevent blood from interacting with your underlying solid. It'll just kind of penetrate that water film and then get in there. And with some other liquids, let's say it's oil, you really have to start playing with the energies of the system that you're designing to make sure that you're able to trap your film that's desired, and being able to repel, like the underlying surface should repel the thing that is not desired. And if you don't match this properly, let's say if the thing that you want to repel likes the surface more than the water does, or whatever liquid that you trap, then it can displace that liquid, and essentially you get pinning, and it's even worse than if you didn't do anything. So kind of some basic design tips that we're at right now is that if we have some texture, the smaller the capillary force, we're able to change the wetting properties to kind of go in more extreme in whichever direction. If we control the surface chemistry, this functionalization, then we can start to change the wetting, the contact angle of these systems. So if you go from, say, something like Teflon, maybe in your nonstick pans, to something that is hydroxyl terminated, then you can drastically change this the surface chemistry and the apparent wetting. And if we design it properly, we can have a functionalization that will really trap the lubricant, what I'm going to call the lubricant, or this liquid layer, and will really repel whatever it is that we're trying to repel. So it's kind of having all three of these can really help us design a stable surface. To walk you through some examples of what we did in the lab is one particularly useful case maybe on stainless steel. Stainless steel is a material that we frequently use in the world and has desirable properties on its own. So if we can add certain properties, like not being able to, like for blood not to be able to attach to this, then maybe we start to get some benefit. In this case, we did an electrodeposition of some nanostructures. This is tungsten oxide, so something that's very hard that will be robust and kind of resistant to scratches and damages by touch. And we can conformally coat stainless steel objects. And when we use this in application testing, so I gave you the example of blood, we could see that when you have a control scalpel, if you dip it in blood, when you remove it, there's still some blood that's sticking onto that scalpel. And this is just kind of visualized in the washing in just some water. If we have a super hydrophobic surface, which is this second case here, then you still do get some blood attaching. It's less than what you got when it was just untreated, so just hydrophilic steel. And when you have a slippery coating, which is this third example here, this slips, you get very, very, very little blood to be attached to your scalpel. So this may be useful in trying to just keep objects clean, and maybe also in bacteria. So here we incubated these treated scalpels, into a broth of culture medium, Staph aureus, so something that's very medically relevant. And we see that we get about like a 5x reduction in adhesion to the slips compared to the control surfaces. So we could start to get, this is a really high concentration exposure, but we can start to get some biological resistance through this design strategy. To kind of summarize where we're at right now, we're dealing with biological organisms. Many times they excrete some sort of adhesion promoter or some sort of adhesive, and they make contact with a solid surface that they can then kind of grow onto. What we're trying to do here with this case is trying to prevent the solid contact. So just have that organism or the biological fluid only interact with the liquid layer and not the underlying solid. So to try and mask that solid layer. Another strategy of doing this, previously I was just showing you nanostructures, but the structure, the nanotexture isn't necessarily required if we start to design our systems kind of a little bit more intelligently and elegantly. One way may be to have the lubricant penetrate completely into the coating, and not, have a thicker coating, not just a thin little film. And we might be able to do this by infusing this lubricant, this liquid, into a similar polymer system. So to have like this matrix, and then to have this liquid that impregnates the matrix and then presents at the surface. So we can do this, we have shown this through using PDMS, so using these silicone systems where we polymerize them to form the actual polymer network. Then we add in the oil, which then intercalates into the polymer network. So we call it swelling or infusing. There is this dimensional change that happens. But at the end of that dimensional change, there is a lubricant layer that is presented at the surface. And the benefit of this, this approach is if that top lubricant layer is lost, that you have this reservoir in the polymer matrix that will then continue to replenish as there is loss. So this is all kind of thermodynamic design. We're trying to figure out like, how can we get the lubricant to be stable on the top surface? This is something that's pretty interesting, and there's a lot of interesting physics that goes into this. That's still kind of evolving of trying to do this with not just PDMS, which is great for some medical systems, but other materials as well, to just have let's say this 2D slippery coating rather than this nano-textured or structured coating. So to take this silicone example with PDMS and apply it to bacteria, again, using Staph or Pseudomonas, we can have this culture that's being pumped through a tubing, a silicone tubing, and half of it we've treated, half of it we haven't treated. And then after it's been pumping for a week, we can look at it. In this case on the right here, we can stain it with crystal violet to just roughly at a macro scale see what is sticking, what's not, and where. And we can see that on the treated side, where it has been swollen with the lubricant, that you don't get bacteria to be sticking to the surface. And on the untreated, you end up seeing heavy sticking. So in the confocal here is just kind of this Z stack to kind of show you the same thing. So you do kind of get these little points, maybe those are defects on this C and D example, where you do get some spot pinning, but drastically reduced from the whole sheet that you form when you're untreated. So, that's just if you want things to not stick from the very beginning. But now that we're starting to approach this and we start to have this in like as an approach in our design kind of like toolbox, we could start thinking of doing these combinations of stick and non-stick systems. Say in a case where you want to grow a sheet of cells in a culture, you need some sort of adhesion promoter, so maybe fibronectin that will signal the cells that it's okay, that they can settle on the surface. And underneath that fibronectin film, we may have a lubricant retaining coating. It's not so actively slippery when we apply the fibronectin, we apply the cells, but after the cells have formed, they form their sheet and we want to harvest them. One approach would be to inject lubricant underneath the cell sheet and allow that to interact with the lubricant retaining coating, become slippery, and then release the whole sheet as a whole, which can then be harvested maybe through transfer, maybe on a piece of filter paper or something, and then you can do with it whatever you wish. So this is an interesting strategy and an interesting, again, going back into learning new strategies and learning how we can start to approach this. In the future, something that will be coming out in publications, we've been doing a lot of testing around the world with this silicone coating, this infused silicone coating, and trying to see, can it be turned into a marine coating? Can it be put on the bottom of ships to save energy through roughness, and also to reduce the amount or prevent toxic materials from being used in these coatings? That's what's typically used to just kill whatever sticks or whatever touches. So we're seeing really promising results. We had a publication in Science that showed when muscles stick to the surface, that you essentially you don't even have molecular signatures of the muscle adhesive on the surface. So very repellent surfaces. So now we'll be moving into the getting into stripes and patterning, which stripes and patterning is something that you see, throughout the animal world, frequently in skins and hides. And it could be stripes, it could be spots, it can be these sort of more complex patterns that you see. One thing that is in common in some of these cases is that you get this characteristic length scale of these features. So in this case, I'm calling the yellow phase. If you look at it, it kind of has the same size, same width throughout the animal, pretty much no matter where it is. And then the black phase, it undulates a little bit, but you kind of get this average sizing. And there's not I guess there's not good agreement as to the exact mechanism of how in biological systems that this is created. But one explanation that seems to match pretty good is this reaction diffusion model. And in a reaction diffusion system, you have this notion of these two species. You can call them AB, or activator and inhibitor. The activator produces more activator and more inhibitor, so it produces kind of more of everything. And the inhibitor tries to reduce the production of everything. So you have these two competing forces. And if you start to map them out and kind of vary the parameters of the activator and the inhibitor, you start to get this phase diagram where you see these opposite phases, extreme opposites, from the gray to the white on one hand. And then you can start to have these spots or inverse spots, or in the middle, this labyrinth pattern. So this kind of starts to match up with what we're seeing in this pufferfish that we're looking at. So to me, the labyrinth pattern is something that's interesting. It has a really high surface area, and sometimes surface area can be really valuable, say if you're designing a battery or a number of other systems. So this essentially is like this balance of two forces. And one system that we can use to easily start to cycle through these different phases that we find is through a magnetically responsive liquid called a ferrofluid. Ferrofluids, if you've seen it before, most times you see it in this spike formation. And the spikes will form perpendicular to the magnetic field, to the magnet. And you have the magnet down here, ferrofluid above. And as you get it closer to the ferrofluid, you're going to start to see these spikes grow. And all of this is essentially this balancing act of these forces of the surface tension. So in this case, it's the ferrofluid and the air, the gravity, which is trying to push everything down. So that's going to be the resistance to building really tall spikes, and the magnetic field, magnetic force, which is trying to promote this growth of spikes. So if we were to liken it to the inhibitor-activator model, this reaction diffusion system, the surface tension and the gravity are the things that are trying to restrict growth, and the magnetic field is the thing that's trying to promote growth. And this down here is just trying to essentially say that this is at equilibrium when the sum of the forces is equal to 0. So similar to this contact line argument that I was making earlier, of this balance of forces is when you get things to be at equilibrium. So Fg here is the gravitational force, magnetic force is m, and surface is s here. So just a different schematic and done in a slightly different way. We have the magnet here below in E, the ferrofluid in D, and now we're starting to add this casting fluid. This casting fluid is immiscible, and you notice that even with the same magnetic field, you go from spikes into labyrinths. So this can be explained by looking at this factor down here, which is the surface tension, which is what we talked about in the previous case. And the surface tension, instead of it being ferrofluid and air in the spikes case, this X, it would be ferrofluid and the casting fluid. So this surface tension is what was changing when you add in this casting medium on top. So by changing this ferrofluid, by changing this interfacial tension, now you're able to change the area. So everything else is staying the same, the gravity, the magnetic field, but the area and the surface tension is what's changing in this case. And if we start to move the magnet up and down, we start to be able to cycle through a wide range of different patterns that we can form. And trying to explore these patterns and trying to explore a way to manipulate them, we ended up using this array of magnets rather than having one single magnet. So now being able to have finer control and shaping how the magnetic field looks like in this casting medium. And we're able to actuate them up and down individually using a computer controlled system. So an example of just kind of like looking at this and seeing what can we do just as a quick demonstration, something we wanted to play with was opacity. So can we control how transparent these casted mediums are in certain areas? And one example image that we were using, this is something popular at Harvard's Graduate School of Design. They like using Marilyn Monroe's image for some reason, but taking that image and pixelating it and bringing it into essentially trying to break it up into these individual tiles that we can then cast through this programmed stepwise magnetic control. So we're able to start to get this gradient of magnetic field across our tiles. And here we're just using epoxy resin. So this is something that's clear, pretty easy to work with. And the final result, you may have to kind of like squint at it a little bit, but this is about a 1/2 meter by 1/2 meter tiled Marilyn Monroe face. So this is just a demonstration, and this is an epoxy. We can start to change the casting medium that we're using, maybe to something a little bit more architecturally relevant, like cement or terracotta. And again, this gets into each of these will start to change the interfacial tension, just like how adding the epoxy changed the pattern that we formed. So by cycling through these materials, you're able to change the interfacial tension, which then also not only changes the area, but also changes the characteristic length scale that you're forming these features at. So this is kind of just starting to explore this space. And in the future, if anyone has any potential applications, maybe it's through cell culture, maybe it's for sensors, but I'd really like to explore this. Feel free to reach out of how we can apply this same type of physics into these new systems. And really quickly, this will be a little bit more brief than the others. We'll go into color, structural color, and optical interfaces. So the previous cases were all dealing with these interfaces of sorts, mostly dealing with liquid interfaces, liquid-liquid interfaces. And now we're going to be thinking about optical interfaces. So structural color is something that, whether we know it or not, is kind of all around us. And maybe most of us see it in soap bubbles, but you may see it in nacre, in seashells, or you may see it in the signaling that for a bird, to attract a mate. In this case, this berry is trying to get eaten, so it wants to attract some birds that will then eat it, and then move the seeds to another place and defecate and let that plant grow there. In the case of the beetle, it's about thermal management. These beetles exist in really hot places. So how can it control how much light is being absorbed into its body? If it can find a way to completely reflect all the light, then the internal temperature can stay far cooler. So, getting into applications that humans may be interested in, it may be in light harvesting, which is just solar. It may be thermal management. So maybe you want your building to stay cooler and reflect all this infrared light that's coming from the sun. It may be simply a special effect. This is chocolate down here. So maybe you just want to have some nice, fancy special effect chocolate. Or we could start thinking of how can we use this concept in more interesting and applied ways that really tell us information about a system. So to get into the difference of essentially structural color versus pigmented color, in pigmented color, frequently the color is from the absorbed light that's happening, that's incident on the pigment. So you have some chemical molecule that will selectively absorb certain colors based on the structure of that molecule. And we see this kind of, this is the most common thing that we experience when we're thinking about color. So this is subtractive color. In nanostructures, or this structural color, we're dealing with additive color. So it's selective reflection of colors. So instead of it being absorbed, it's being, the length scales of these features are just the right size where they're just reflecting certain colors. And you can do this in many different ways. You can have a single film, and this is all about refractive index differences and the thickness of these different layers. I won't get into the math too much, but it's really interesting to dive into this and all these different strategies. So you can have a single layer, you can have these multiple layers, and the multiple layers is something that we'll be focusing on for today. So in the case of the berry, if we start to zoom in on the berry, we look at the skin, just cross-section it, and look really, really closely, you can start to see you get these coils that are forming. And the coils are made up of one material, but on the inside of that material, similarly in nacre, you see this thin protein layer. So you have these two different materials, actually. The protein is likely templating this growth of this optical material. And you get these kind of rolled structures. And that's a way that you can get a lot of layers really quickly. We see this in other systems, and in this system particularly, we see this blue-green scattering or reflection of light. But in other systems that we're used to, maybe you're making croissants. And the way that you make croissants is, maybe it's a little different than how the plant makes its layers, but the end result is pretty much the same, is that you end up getting, you want this interface of batter and butter, and kind of rolling it up is a way to quickly exponentially grow the number of layers and make it nice and flaky. If we start to think of nanostructuring and nanofabrication, we can control layers really precisely using the tools that we have. And in this case, we're dealing with just two polymers, so PDMS and this PSI, PSPI polymer that we can coat, just in two dimensions, and then roll it up, just like how we do with the croissant. And you end up getting these cylindrical multi-layers that are elastic. So these are, this is a polymer, this is flexible, it can bend, it can stretch, and you do get color, selective color reflected back. So in the case of the system, we could start to think of it almost like as a strain sensor, whereas you start to stretch these fibers, the balance of conservation of mass, everything needs to, if you stretch it one way, then things need to contract in the other way. So the spacing of these are starting to decrease, which will then start to shift the colors that will be reflected from these fibers. So this is something that's pretty interesting, and just a way to start thinking about how can we start designing smart materials to be able to tell us more information about what's going on than just simply a static color. Let's say if you put a pigment in there, it can only be blue or it can only be red. But now let's say if you have a band that you're stretching for physical therapy purposes, maybe there's a certain color that you want to reach. So you need to move your body a certain amount in order to activate that color. So we could start thinking of how we can design interesting products this way. And this is an area that I'm also very interested in and will be continuing to work in, of trying to combine these worlds of nanofabrication and digital fabrication to design more elegantly, as well as to be able to design at larger scales. Nanofabrication is great for computer chips, but if you want to wear something, let's say, that has structural color, nanofabrication is maybe a little bit difficult in that regard. So to kind of wrap up this presentation, I know it's like a whirlwind drinking from a fire hose, but hopefully you were able to glean some new strategies for designing or applying wetting physics to systems, and how we can start to explore this stick and slip kind of phenomenons to get things to solve problems that we want. And in the case of patterning, I think we need to get away from, as a whole in manufacturing, get away from precise control and precise deposition of patterns and start to move towards self-assembling patterns, or maybe self-organizing systems. So in this case, in the case of the reaction to fusion, this is through competing forces. So how can we use competing forces to our advantage here? And then in the case of the berry, we're getting into non-pigmented color. So this has a wide range of applications, and maybe we're not dealing with color specifically. Maybe we're dealing with infrared or some other wavelengths that may be of interest. Say if you want to wear something and you don't want to get hot. So this is just like a brief overview of of a bunch of projects that I've worked on in this space. I have worked on so many other spaces as well, not just the bio-inspired materials world. But feel free to check out my website if you want to know more about some of the things that I've done. And feel free to reach out to me if you have ideas that you would like to collaborate with. I'm pretty observational, and I really learn from observation and can apply analogously problem solving techniques from one space to another space. So feel free to reach out to me. And with that, I will move towards the questions.
Thank you very much, Onye. It was a great presentation. It was like an open mind presentation, because as a scientist here in Guatemala, we don't work that much with engineers or with designers. So we have a question in our chat, and it is from Karen, and she says, do you think it's possible to change in the future all the design of the synthetic materials to a biological design?
So I think, in my opinion, it's sort of like this false dichotomy. I think nature has a suite of materials and a suite of chemistries that it can deal with. And man-made objects, we have a much bigger range. So I think there's really benefit of both. It depends on the use case. But if you're going to be dealing with high temperatures, or you're going to be dealing with, let's say, caustic materials, you really probably don't want to use some biological materials, but there are definitely many cases that we can switch over from synthetic materials to more biological materials. So hopefully that answers your question.
Thank you. And another question is, what kind of disciplines or professionals has to meet together to develop this kind of project that you present?
Oh, that's a really interesting question. I think for me, I found material science at kind of towards the end of my undergraduate education, and I found it as an extension of chemistry. So material science kind of sits in the middle of touching all these different fields. Some projects that I didn't present on were about cement and building materials. So you can imagine civil engineers are pretty interested in that. Maybe you're talking about a space shuttle going into space, has to resist these high temperatures on re-entry. So the aerospace engineers, they'll be interested in this. So I think it's really a way to start approaching problems and a way to start thinking about how to understand systems and how to design better systems. So I think depending on your interest, you may come at it from a certain perspective. For me, I'm interested in just new strategies of how we can design and problem solve, coming from this kind of chemistry, chemical engineering perspective. So, that's kind of what I bring to this. But I'm now starting to interface more with the biological side. So, now I'm doing wine. So, you never know where this will take you, but starting off with where you're at, maybe it's biology and understanding a specific material system. At the end of the day, a lot of this comes down to physics. So maybe applied physics, starting with the basics and then learning new ways to apply them is also an interesting way.
Thank you very much. That's the idea with this Congress, that we meet a lot of professional disciplines to work together. It was a great presentation. I really love it. So I think if anyone don't have any more questions, Luis, it says that great presentation, very interesting topics. So thank you very much, Onye, for your time. We are going to keep in touch to talk about projects. So thank you.
Yeah, and thank you for having me. I look forward to seeing what else is presented in this Congress.
Oh, thank you, Onye. See you. And for the rest of the people, we are going to now to present the poster competition that is coming next in 15 minutes. So join us for the presentation of the video pitches of the team. Thank you very much and see you in 15 minutes.